So Streptococcus is also gram-positive pathogen. And in this word, streptus, it's also from a Greek word. It means either easily bent or twisted like a chain. So it's like a chain or like a rope. So twisted chain. So Streptococcus are a line of cocci. They line up together to form a chain. So that's a very special organization, special grouping, the word we learned on the first lectures. And they are also important pathogens in human society. And we'll learn their classification, and we'll learn their colony morphology and hemolytic reactions on blood agar. And we'll learn their serologic specificity. That's because of the cell wall group specific substances. Actually, they are polysaccharides. And also we'll learn their biochemical reactions. So that based on these different reactions or different methods, we can derive useful information for diagnosis. First, we learn hemolysis. Hemolysis here in this word, hemo means blood. Well, it actually means red blood cells, because at the beginning, when people get to know blood, people only know the red blood cells in it, and they don't know the immune cells, lymphocytes, and other cells in the blood. So lysis means lyse, so you break it up. And hemolysis, it means the lysis of red blood cells. There are different types of hemolysis. The first one is beta hemolysin. So that's the enzyme or the toxin which can lyse red blood cells completely, like in this corner. They can lyse the red blood cells completely, so that in this area there is no red blood cells left. It will clear the zone due to complete disruption of the cells. A second type of hemolysis is called alpha hemolysin. So those are the hemolysin which can incompletely lyse the red blood cells, and results in the reduction of hemoglobin. You know, we have in the hemoglobin there is ferric iron, and the alpha hemolysin can reduce the ferric iron to ferrous form, so that they will form the green pigment on the agar. So if you notice the formation of green pigment on the agar, that is alpha hemolysin, due to alpha hemolysin. Another type is called gamma hemolysin. Gamma hemolysin, it actually doesn't lyse the red blood cells. Like this on the top, you cannot see any change in the agar. They are not hemolytic. But I don't know, because I don't know why they got the name of gamma hemolysin, even though they don't lyse red blood cells. Yeah, I'm wondering why. I'll check some information later, or you can read the textbook or check it online and figure it out. Even though they don't lyse red blood cells, why they got the name of gamma hemolysin? Okay. So this is a hemolysis. Second is that a group-specific substance. So this is a criteria people used to classify Streptococcus. This criteria is called Lancefield classification. So that is based on their surface antigens. You know, they have surface carbohydrates, and based on the type of surface carbohydrate, they can classify Streptococcus in different groups. Right now we have 17 different groups with letter name, like group A to H and group K to U. In each group there are one or more species. Okay, in each group there are one or more species. The most important one are group A, B, and D, which are important human pathogens. While in group A, 90% of human pathogenic human disease is due to group A. So it contributes 90% of human pathogenic streptococci. So we'll focus on group A for sure because they are the major ones. Okay, the first pathogen we'll introduce is the species called Streptococcus pyogenes. Sometimes it's short for S-point pyogenes. And they can cause infection in the throat. And okay, so this is the first pathogen we'll learn. Let's see. And their morphology, as you can see in this figure, this is the morphology after gram staining. You can see that they are showing this purple or blue-purple color, and that means they are gram positive. And they form these chains, long chains usually, but sometimes they can also form pairs. So maybe the chain will grow longer. Yeah, pile means pus. Yeah, and they produce capsules actually, and the capsules contains hyaluronic acid. So let's see hyaluronic acid. And they are catalase negative. So this is the model structure of the part of the bacteria. You have the membrane, that's the double layer lipids, and you have a thick peptidoglycan, that's the cell wall, and from the cell wall there is a long projection, the long hair. This long hair is a M protein. It projects from the cell wall. Pyogenes can produce M protein, which is a hair-like long projection from the cell wall. And they can also produce toxins and enzymes. The first type of enzyme is called streptokinase. Streptokinase can transform the plasminogen of human plasma into plasmin. You know, plasmin is an active proteolytic enzyme that can digest fibrin and other proteins, allowing the bacteria to escape from blood clots. You know, maybe your system can respond to Streptococcus infection and form some blood clots, so that you can the immune system can lock up the bacteria and can hold the bacteria there. But the bacteria can produce some kinase, streptokinase, and manipulate plasminogen into plasmin, and plasmin can dissolve the clot, dissolve fibrin in the clot, so that the bacteria will be released. They can escape from the clots. It's a very special, a very clever strategy. They can also produce streptodornase. That's the enzyme which can degrade DNA molecules. We mentioned that DNA molecules are long molecules. They can trap the bacteria. They are sticky. They are gel-like, glue-like molecule and trap the bacteria in the gel, but they can degrade the DNA molecules so that can help the bacteria to escape. Okay. And other toxins and enzymes, they can produce hyaluronidase. So that's the enzyme we introduced just now. Hyaluronidase can split or catalyze the hyaluronic acid. This is the important component in our connective tissue, and if they can hydrolyze the hyaluronic acid, they can help the bacteria to spread. They can also produce pyrogenic exotoxins. They mediate the pyrogenicity. It has been associated with scarlet fever and streptococcal toxic shock syndrome. We'll introduce later. And hemolysins. That's the toxins or enzymes which can lyse red blood cells. There are two different types of hemolysins. The first one is hemolysin O. Hemolysin O is it's a protein hemolysin, and it is antigenic. It can stimulate the human immune response to produce anti-streptolysin O, short for ASO. So it can stimulate the immune system to produce antibodies. So it is antigenic. But streptolysin O can be rapidly inactivated by oxygen. So it's not stable in the presence of oxygen. So this is the first one. Another one is streptolysin S. Streptolysin S. Different from O, the streptolysin S is stable in the presence of oxygen. And streptolysin S is responsible for the hemolytic zones around the colony. It is stable with oxygen. But keep in mind that streptolysin S is not antigenic. It's not antigenic means that it does not stimulate the immune system to produce antibodies. So you cannot use antibodies against streptolysin S to detect this antigen. For clinical findings, so that's what we will learn, what type of disease they can cause. They can cause disease by invasion of the bacteria or beta hemolytic of group A bacteria, or there are disease by local infection with asparagines and their byproducts, and they can invade. The group A bacteria can invade and cause infection, and which can further cause like streptococcal toxic shock syndrome and scarlet fever. And these are disease caused by bacterial infection directly. And there are also another group of disease caused after bacterial infection. This is due to the immune response. So after the bacterial infection, even though the bacteria are cleared out, the immune response can produce some molecules and can cause damage in the host. And this type of disease we call the post-streptococcal disease. And we will introduce these different infections, different disease one by one. The first one is caused by like invasion by the bacteria or hemolytic results, like this one, infection on the skin, or cellulitis, also infection in the skin, and this will cause a large area, and the spread is rapid. It can spread from one one spot to a large area. And also another one is called puerperal fever, and this is the major threat in the history for human. So that's when women give birth to babies, and the Streptococcus can cause infection in the uterus, and that is a very big threat, because in the history for a long time having a baby, give birth, is not an easy task, and a lot of people, a lot of women, a lot of moms got infected and cannot survive. And right now with like a hygiene technique and the modern hospital equipment, this is not a big problem anymore. And on our first lectures, when we talk about asepsis, we introduced the first pioneer in decrease the rate of infection. So that is at the early 18th century, before the setup of microbiology. And based on his work, later Joseph Lister developed and spread the technique of antisepsis, the asepsis methods. Okay, a second group of disease is by the local infection of the bacteria and their byproducts. Like they can cause a sore throat, so that's an infection in the throat, and also streptococcal pyoderma. This is the local infection of superficial layers of the skin. Okay. Because we have so many points, I didn't prepare slides with pictures. So maybe you can find some picture in the textbook, or because you have internet, you can search those terms and find the proper picture images to help you understand different type of infection. Okay, another group of disease is due to invasion by group A Streptococcus. They can lead to the streptococcal toxic shock syndrome, including shock, bacteremia, respiratory failure, and multi-organ failure. And this is the toxic shock syndrome. And also scarlet fever. This infection results from this disease results from pathogenic exotoxins in association with asparagines. This shows a rash on the trunk after 24 hours of illness and spreads to involve the extremities. So this is scarlet fever. I should have prepared some images here to show you different types of infections. Okay, the last one is the post-infection disease. That's the post-streptococcal disease. This is because that in the Streptococcus pathogen, they somehow evolved some epitopes which are similar to human epitopes. So those epitopes can stimulate the immune system to respond, to produce antibodies. And that antibodies will cross-react with components on the human tissue itself. That's kind of like an immune system attacking the host itself. So that will lead to different type of disease. For example, the antibody-antigen complex. That antigen is from the bacteria. Antibody is produced by the immune system. This complex can precipitate in the basement membrane and then can cause some damage. This is one type of post-infection disease. Another one is called the rheumatic fever, and this is because that the bacteria share some similar epitope with the tissue of human heart, on the heart muscle or the heart valve, and infection of the bacteria will lead to the immune response, and the immune response will attack the heart or the valve and results in damage in these parts. And it can also cause fever, malaise, and also other symptoms. Okay, so that's the clinical findings, the disease they can cause. For diagnosis, for like laboratory tests, we can perform according to the standard procedure. First, we collect specimens from either the throat or the pus or the blood or the serum. So we collect different type of species, and in different type of species, we can detect different components. For example, the smear. We can check the smear with a light microscope, especially after staining. We can culture the smear to see whether there is live bacteria. We can use blood agar plates. That's the agar plate contains fresh red blood cells. Or we can run test their hemolysis activity and see whether they can lyse the red blood cells in the blood agar plate. We can also detect antigens. That's the ASO test. And also we can, detect the antigen is not the ASO test. We can use known antibody to detect the antigen from the bacteria. So that's also the antigen-antibody interaction. Or we can use the ASO test to detect antibody in the serum. So this is a serologic test. We can use specific antigen to detect whether there is antibody in the patient's serum. And this is the ASO test. And if the patient's serum can react with the bacterial antigen, we'll know that there is some antibody, and you can get some information. For bacterial treatment, and of course, this is a gram-positive bacteria, and they have a thick cell wall, and we know that some antibiotics targets on the cell wall, so they attack the cell wall to inhibit the bacteria, including penicillin antibiotics, and or its derivatives. And penicillin G is the first line of antibiotic to treat Streptococcus. And also, you might use other antibiotics if the patient is allergic to penicillin. Okay, but we should keep in mind that the antibiotics can only inhibit the live bacteria, can only kill the live bacteria. If the disease is due to the post-infection response or the post-infection immune response, the antibiotics does not help. Okay, so that's the first pathogen we introduced, the Streptococcus pyogenes. Okay, another one is Streptococcus pneumonia. This is also important because Streptococcus pneumonia can be a big threat for some group of people. For example, those very young newborn babies or very old, the elderly population, they are more susceptible to this pathogen. Okay, first we want to examine their morphology. They are gram-positive for sure, and but even though they are they are members of the Streptococcus, they are not forming the long chain like the previous one we learned. They are only forming this diplococci. Diplococci means two bacteria. Di means two. So usually there are two bacteria lying together. So this is a pneumonia. And they can produce capsules. And even in this gram staining, you can see the hint of capsules. You can see something around the bacteria. And you can culture the bacteria on blood agar plate. And this is same to the first pathogen we introduced. They produce alpha hemolytic, and we learned that alpha hemolytic is incomplete lysis of red blood cells and can form the greenish pigment. The bacteria can produce autolysin. This is a very interesting property. Autolysin means that the bacteria can produce something which can lyse themselves. They can kill themselves. By killing themselves, the bacteria can recycle the material in the community so that they can divide and grow and generate more cells. So when you culture the bacteria on the plate, the colony will look interesting because in the center of the colony there is a depression. So in the center of the colony, its system cannot destroy the bacteria immediately. So they can inhibit the ingestion by phagocytes. So they inhibit the phagocytosis. They produce pneumolysin, and this is the lysin which can destroy red blood cells and can inhibit lymphocyte proliferation, can inhibit neutrophil chemotaxis. And they have surface protein for adhesion, and they also produce protease which can degrade IgA. And you know that we have mucous immune system, and we have a mucosal membrane, and in that mucosal membrane there are a large amount of IgA which is important for defense. Meningitis, that's an infection in the brain and can cause endocarditis. So that's the infection in the inner part of the heart, and can also cause septic arthritis. So that's an infection or inflammation in the joint parts. A different disease, that's a different type of disease. For clinical of diagnosis or laboratory tests, we need to collect a specimen, and this work can be done in your clinic or can be done by your nurse. In the future, when you are a doctor, you need to collect either the sputum or blood or the spinal fluid. All these specimens you collected should be sent to the lab immediately. That is because this bacteria can produce autolysin. if you leave the specimen in your clinic for like a long time, and during that time the bacteria will kill themselves, will lyse themselves. So you need to send the specimen to the lab immediately. And in the lab, the people working there can first check the morphology, whether it's gram-positive or not, and they will find the diplococci. And meanwhile, in the specimen, they will find like a lot of neutrophils and many red blood cells. That's the characteristics of infection by this Streptococcus pneumonia. And they can run the capsule swelling test if you submit the patient's serum or blood, and if the serum mixed with the bacteria can cause swelling of the capsules, they will know what type of infection it is. And for culture, they can isolate the pathogen by culturing them on the plate. And again, people use blood agar to culture the pathogen. And on the blood agar, they can form alpha hemolytic zone. So they will cause the production of green pigment. So that's the alpha hemolytic zone. For treatment, again, penicillin G is the best choice. And if penicillin G is not working, if the bacteria are resistant to penicillin G, you can use like other susceptible antibiotics such as vancomycin to treat the bacteria. Right now there are 2 different types of vaccines available, which can give to the high-risk individuals, including the older ones or the very young ones. So it will give them protection. A second type of vaccine is called conjugate vaccine. This conjugate vaccine, they actually combine the polysaccharide with diphtheria protein, so that this polysaccharide will bind to the protein molecule. And because there is protein molecule, it is more antigenic. So this vaccine is more powerful than the polysaccharide itself. So that you can give this vaccine to the very young kids, the newborn, from 2 to 23 months. So 2 months after they are born till they are 2 years old. During this period, the polysaccharide vaccine does not work well, but you can use this conjugate vaccine. Okay? And I guess in your family today, we covered 2 different groups of gram-positive pathogens, Staphylococcus and Streptococcus. And in each group, we introduced a couple of important pathogen species. Here I have some video. I think those videos should be available online. If not, I'll check later if those videos